All right, so today I'm going to go over another heavy protein genetic type of paper titled Proteomic Biomarkers of KLS. Um, it's a pretty complicated paper, but again, my goal is to um, provide insight into a lot of the jargon and share what the authors are saying, maybe in more of a layman's terms. All right. Um, so the goal for this paper was that we know that there's no biomarker for KLS. Some have been suggested, like hypocretin or orexin because of its association in things like with narcolepsy. But really a big profile picture of what could be upregulated or downregulated in KLS patients was not done prior to this study. So this tries to fill that gap and also provide a launching pad, hopefully, for more. All right. So intro, like most papers, it's describing what KLS is a little bit clinically. Also provides a little bit of background into some of the hypotheses that are talked about for causes, namely one of them being autoimmune or infectious association, because a lot of episodes begin with some sort of infection, flu-like illness, something, right? A lot of people get triggered by one, and there have been many cases that have they started to experience KLS at all following some type of infection. Genetically, there was the TRNK1 signal region locus of the genome that had a higher association, but specifically with respect to a stronger association with those that experience a difficult birth. I did do 2 videos on that, one on the TRNK1 portion specifically, and then one on the gene environment interaction model, followed up with that. So those are 2 previous videos. Highly recommend you watch those. And the neuroimaging studies to suggest that there's changes in metabolism and perfusion in the brain. But again, the goal of this study is to provide a high throughput approach to screen a ton of different proteins in the CSF, cerebrospinal fluid, and serum, looking for any signatures, I guess, that would help distinguish between KLS from controls or provide some insight. And a large part of this paper is describing a lot of methods. All right, so the type of techniques that they used, right? So I'm going to do my best to describe those. So the SomaScan assays, all right, that's a type of technology that can measure hundreds, thousands of proteins simultaneously from a given sample of fluid, and so it takes synthetic pieces of DNA engineered to bind to specific proteins, almost like a lock and key. And so when it binds to that target, it generates a signal, and that signal can be measured, right? And so in this study, CSF was profiled for, 1,133 proteins in the training set, and then 615 proteins for validation, and then in the serum profiled for 1109 proteins in the training set, and then validated thereafter. All right. And so that's just describing the type of assay that they did for screening. All right. Study cohorts, all that's kind of describing is that the way that they picked and chose cases, right? So I believe they describe All patients were diagnosed using the known criteria at the time. It has been since updated since this paper. But in CSF, I believe it was about 30 KLS cases and over 100 controls, which is good. And then in serum, over 26, I believe it was 26, KLS cases, and then about 65 controls. 11 KLS cases, they used both their CSF and serum that was available. All right. Then some matching. Because KLS patients and controls differed in age, sex, and things like BMI, that's just some criteria that's often used. Researchers use case control matching. All right, that's what this means down here. And they used a match it R package to do that. I'm a little unfamiliar with what that package is. However, I'm assuming it made their life easier. But the goal is to pair each patient with a fair matched control with demographics, sex, BMI, things like that. So to kind of reduce the confounding variables that are involved. So that's to describe how they matched up some cases. All right, some statistics. I hate statistics, so I'm going to do my best to describe some of this. So a univariate analysis, that means examining one protein at a time, univariate, all right? They used the log2 transformation. What that means is, I think that's in the first sentence. So raw protein measurements are then converted to a scale score so that you can compress extreme values in data to more manageable numbers. That's really all that means. Taking a logarithmic approach to use better numbers that are more simple to the eye. Principal component analysis, that's in which it's basically because you're measuring so many proteins per case, the PCA simplifies that complexity by finding the main axes of variation, which is like taking a 3D object and finding the best angle to photograph it so you can see the most important differences in a 2D picture. So that's the check almost for technical problems to see if the KLS and control samples naturally separated. All right. The Mann-Whitney sum-rank test, that's a non-parametric statistical test that basically it's a test that does not assume the data falls below the bell curve. Some of us may remember that way back in high school. And so it ranks all the values from highest to lowest and basically asks, are the ranks of KLS group significantly higher or lower than the control group, right? P-values, we've all heard, most of us have heard of p-value before, and so that's just a threshold to answer the question of if there are basically truly no difference between KLS and controls, how likely would it be to see a difference by chance alone? And so a very small p-value means that that difference is very unlikely to be due to just chance, which we would call something statistically significant. Right. The Benji-Hock procedure, that in the FDR corrected p-values. So when testing over 1,000 proteins, some will appear significant purely by chance, right? And so that type of statistical procedure kind of adjusts for that by converting the p-value that we just mentioned before into q-values that they call a false discovery, right? And so a q-value of a certain threshold or below a certain threshold means that all the proteins that are called significant are expected to be false positives. And so that takes that false discovery, meaning a false positive, is essentially into consideration. And a log fold change measures the magnitude of difference. So a log FC of +1 means that there is a twofold increase, or -1, positive or negative, would mean that there's a twofold lower. So instead of taking this stream of numbers of this complicated, this upregulated versus this downregulated, you can convert it into a logarithmic portion. That means if something is 0.1 positive 0.15, then it's upregulated by that multitude higher, and same with lower. All right. The pre-processing of the multivariate analysis. All right, so while the univariate analysis looks at one protein at a time, univariate analysis looks at many proteins altogether to try to find patterns. All right, and so keeping only proteins measured in both training and the validation panels, what they did was remove batch effects. So there's sometimes like systemic technical differences between the SomaScan runs when it's screening. And so it's almost like calibrating two different thermometers so that their readings are comparable. And so, yeah, that was their pre-processing for scanning a bunch of different proteins together. Lasso classifiers. So there's a bunch of different numbers in that and what that means, but that's basically a machine learning algorithm that builds a mathematical equation combining multiple protein measurements to predict whether a sample comes from a KLS patient or a control. So the lasso effect, or that part, kind of forces the equation to use as few proteins as possible, meaning the unimportant proteins drop to 0. So that kind of prevents overfitting proteins and identifies the most important ones that fit within the lasso, so to speak. All right. Bootstrap. All right. All that means is because lasso can select slightly different proteins each time that it's ran. What they did was they ran a model 1,000 times, where it says here most occurrences across 1,000 models. All right, so they ran the model 1,000 times on slightly different random resamples of the training data. And so proteins selected in over 70% of runs were considered robustly predictive. And so later I'll describe a finding that would describe something as enriched in that data, meaning that in that 1,000 times of it being ran in that bootstrap analysis, 70% or higher is what they can consider to be crossing that threshold. All right. Tissue enrichment analysis. All right. So basically, they ask the question of, do the abnormal proteins come from specific body tissues? And so using a atlas of proteins, they describe here the human protein atlas. The researchers identified if there's tissue-specific proteins and checked whether or not they're differentially expressed in KLS, if they're disproportionately enriched in certain brain regions or immune cell types. And pathway analysis is pretty similar. And so using a few different databases and softwares that are available, I'm pretty familiar with a couple of these, like String. It's a great software. And so they use the String software along with pathways like KEGG and Reactome to basically map differentially expressed proteins onto known biological pathways. And so like taking a list of broken parts and figuring out which machines that they belong to. All right. So the key takeaway from all those methods is that the study used a rigorous amount of analytics and statistical methods combined with machine learning to validate the results and to try to map to see if there's any pathways that are associated with the proteins that are enriched or upregulated or downregulated. All right, and so differential expression of CSF proteins in KLS cases. All right, so notable upregulated ones. So as we can see, IL-31, TGF beta 2, IGF-1, JAM beta B, capsase 10, IL-27, osteonectin, all predominantly from a macrophage microglial axis. All right, and so just to walk through some of those, and so these are some notable ones. So there were more than those listed in that portion that are upregulated, but those are just the ones that I'm going to focus on because they're noted in that section. And so IL-31, that's a cytokine that's produced in response to, it's basically a neuroprotective effect of microglia. Microglia are the brain's resident immune cells. So like peripherally, we have B cells, but B cells and T cells, but the brain has microglia. And so that's the brain's resident immune cells. And so that's relevant to that type of cell. The TGF beta 2, that's an anti-inflammatory and neuroprotective protein to kind of dampen excessive microglial proliferation. IGF-1, some people are familiar with that, insulin-like growth factor 1. That's a growth factor critical for cell survival, differentiation, things like that. The junctional adhesion molecule B, JAM-B, that's a protein that facilitates immune cell crossing through blood vessel walls. So junctional adhesion molecule, all right? And so if that is disrupted, you can have the blood vessel walls can be somewhat more permeable to things crossing through it. Capsase 10, that's a protein involved in apoptosis or expected physiological programmed cell death. And so that kind of suggests that there might be some sort of neuronal stress going on. IL-27, it's another cytokine. ILs stand for interleukins. That is a type of cytokine. Cytokines kind of fall under the umbrella or vice versa. That does exhibit some neuroprotective effects. And osteonectin, right? That's a protein involved in tissue remodeling and cell matrix interactions, right? So again, so you can see that kind of somewhat has this microglial macrophage type of axis that those kind of are relevant to or associated with. Those are upregulated, right? Meaning higher in KLS. Downregulated proteins, the ones that I highlighted here are LVP, some D double Ks, all right? And so, of course, again, there's more, and they list them elsewhere, but because those are the ones that are mentioned in the section, I'll just focus on those. And so the LPB, that's a protein, lipopolysaccharide binding protein. It's a protein that kind of detects bacterial components to initiate immune responses, right? So LPS is something that a lot of types of bacteria have on the outside of them that will kind of bind to and tell immune cells do something, all right? And so that's what that binding protein is on the receiving end. And so it detects if that's present. The D double K proteins, all right? So DKK4, 1, 3. All right. Those are inhibitors of a pathway called the WNT slash beta ketamine, right? It's a weird sounding pathway, but it's critical for axon guidance and dendritic development. And so the processes, meaning the things that jut out to help communicate from neuron to neuron, that pathway, those proteins are involved in that type of pathway that are important for the function of those things. And interestingly, downregulation of DKK has been linked to other conditions of neuropsychiatric origin, like schizophrenia and bipolar. And so these effects tend to be, as the authors say, more pronounced in patients actively in an episode. Let's see, differentially expressed serum proteins in KLS. All right, so the proteins that I just described were up or down with CSF, and now I'm about to describe their section of serum. All right, so they see 117 proteins that are upregulated, 24 that are downregulated, and they mentioned some ones that they wanted to note in this section. All right, so the upregulated ones, like the histone core complex related ones, those are histone proteins that are normally inside a cell nuclei, and the presence in blood at high levels can somewhat indicate some sort of cellular response to damage. Whether or not that's the case in KLS, who knows? FYN and CSK. So FYN, the Circ, I'll just pronounce that. So the Circ family kinases are molecular switches that kind of control or act like a dimmer switch to immune cell activation. FYN is one that activates, and CSK is something that like pumps the brake. And so their simultaneous elevation suggests a heightened, but maybe a regulated immune signaling that is going on. Right? PTP N11 and the PTN6 is a protein tyrosine phosphatases that are involved in immune cell regulation, or the signaling, or the regulation of their signaling. All right, and the tau-to-box binding protein, and so that's a fundamental protein that's on all of our genomes required for gene transcription or turning on the transcription of certain genes. All right. So those that I just described, the authors say were upregulated. Downregulated proteins, some ones that they noted were ENPP7 and interleukin 17 RA1, and some that are further down. And so the ENPP1 is an enzyme involved in lipid metabolism and anti-inflammatory lipid processing. Let's see, where am I at? What's the other one? IL-13RA1. Right. So that's a specific receptor for IL-13, which is involved in anti-inflammatory immune responses. Let's see, was there one more in that section? Yeah, actually, interleukin 1's receptor type 1. That's a receptor for IL-1, which is a key pro-inflammatory cytokine, actually. And so its downregulation may be compensatory, right? So that, let me see. And so that was the differential upregulation, downregulation of proteins in CSF and in the serum, respectively. And so the next section describes an increased cytokine expression in CSF and serum. So this is specifically looking at some interleukins and cytokines. All right. The other ones did include some of that, but this one is just a little bit more specific. So IL-8, 6 upregulated in the serum, CSF, expression of IL-1B, 12 were upregulated in CSF. And so by all means, I'm just going to describe what a lot of these are. And so IL-8 and 6, IL-8 is a chemokine that's basically like a homing beacon that recruits a cell called neutrophils, which is a like first responder for immune responses. All right. It's basically like it's IL-8 is like the gunshot of a it's basically like a firing gun of a horse race. Like everybody start, come here now. All right. IL-6, that was also associated to be upregulated. That's one of the more versatile cytokines in the body. It can act both as pro-inflammatory, as like a alarm signal in certain contexts, but also it can act as a anti-inflammatory mediator in certain contexts. Right? In the CSF, IL-1B, IL-12. All right, so IL-1 beta, interleukin-1 beta, is one of the more potent pro-inflammatory cytokines that's produced. It's usually produced produced by activated macrophages and microglia, kind of acting like a alarm signal that can produce some downstream effects. The IL-12, that's a cytokine that's produced by macrophages, microglia, and dendritic cells that kind of serves as a bridge between an innate and a adaptive immune response. And so its job is to kind of push T cells towards a more pro-inflammatory response, right? Let's see. And so especially with the IGF-1, and so the differential expression of those that I just now described, so it was only like 4 of them, in the CSF could suggest some interactions with cell genesis and myelination. Myelination is a process in which we're wrapping our axons with a insulating sheath of lipid, or microglial activity, right? So the brain's immune cells responding to something, all right? Upregulation of IL-1 and 13, or 4, excuse me, 4 and 13. And so IL-4 is a cytokine that is more on the anti-inflammatory immune response context. It kind of dampens the response, so to speak. And IL-13 kind of works in partnership with IL-4. And so they share a very similar receptor component to activate a downstream effect, kind of reinforcing that there may be some sort of macrophage microglial involvement. But the upregulation of them may be a sign that there's a neuroprotective anti-inflammatory component, kind of maybe in compensation or in response to something, rather than that everything in the brain is just purely destructive, right? And so the brain's immune system could be appearing to actively try to resolve some type of inflammation, not that all these things are the cause of that, all right? And so in a nutshell, right, these cytokine profiles provide a dual-natured response in KLS. There's some that are more pro-inflammatory signals that are elevated alongside some anti-inflammatory or repair signals that potentially suggest that the brain is simultaneously under some sort of immune stress and amounting to like a compensatory response to one another. All right. So hopefully that makes sense. Proteomic based machine learning predicts KLS status, that is. All right. And so that is computer models that I'm going to try to describe. And so the intention there was to build a machine learning classifier to determine whether the protein differences in KLS and controls are large and consistent enough to potentially diagnose KLS from the fluid sample. All right, so they did some models, looks like, with the lasso and the bootstrap, all right, that we talked about before. So they trained the CSF model on 103 individuals. 23 of those were KLS, and then validated thereafter with 61 different individuals, and 7 of those being KLS. And so the bootstrap analysis, let's see. Here we go. All right. Bootstrap analysis identified 8 proteins that were enriched. Remember, I talked about they had that threshold of to consider something enriched being of the 1,000 instances of training, there were over 70%. All right, and so I'm going to describe some of those. They're highlighted in red here. All right, so the TFPI is a tissue factor pathway inhibitor. And so that was at 100%. And so it was selected in every single one of the 1,000 training runs, making it, in the author's view, the most reliable CSF predictor. And so what that protein is, though, it's the body's primary kind of natural anticoagulant, or one of them. So it prevents blood clots, right, in the clotting cascade. So beyond clotting, though, it's also pretty critical for brain blood vessel development, right? Because there's been studies that have shown when you knock that out, there's abnormalities involved in that. VWF, von Willebrand, there's some blood disorders that some people may be familiar with respect to that. That's a large sticky protein inside of blood vessels or endothelial cells, in which it's released when a blood vessel is damaged or inflamed. And so it acts like a molecular glue to help platelets stick together or stick to injured vessels so that a clot can form and go through its healing process productively. Right. And so in the brain, we have the brain blood-brain barrier. And so that von Willebrand factor, the VWF, does play a role in maintaining that. All right. PHI. All right. That is a enzyme that performs a critical step in the production of neuropeptides, like chemical brain messengers. And so there have been so many neuropeptides like oxytocin, neuropeptide Y, vasopressin, are not that functional until PHI finishes its step in the production of those neuropeptides. All right. Let's see. CY is a complement, right? It's a component of complement, and so we have a membrane attack complex with the immune system it's basically like a destructive weapon in response to something. All right. And so C9 is a component of that complement. All right. Albumin, we know of that in some of our blood work. That is a abundant protein in the blood and often acts as a carrying molecule. But at high or low levels, that can influence things from crossing the blood-brain barrier, in which elevated CSF albumin is sometimes used as a marker for blood-brain barrier breakdown, or becoming a little bit more leaky, right? IL-1 we talked about before, LPB we talked about before. So by all means, go back and revisit those in the previous section. All right, and so that was the bootstrap analysis with the CSF. But let me see. And there was also these 5 proteins down here. All right. So in the top predictors, using the bootstrapping, they again observed 5 proteins that were enriched in their training models. And so this is in the serum, I believe, right? Because then that's the context, right? Before we were talking about some serum ones or some CSF ones. Now they did some top predictors for serum. And so ENPP7 is a alkaline sphingomyelin enzyme. That was their most predictive one in their models. And that's predominantly found in the intestine and liver that breaks down something called sphingomyelin, which is a type of lipid fat found in cell membranes and in food. And so beyond that it also serves as a natural anticoagulant, or not anticoagulant, anti-inflammatory agent, all right, which is interesting. FYN, we talked about a little before, that is in the Circ family kinases that is part of the dimmer switches for immune cell activation, right? H2A3, that is part of the histone complexes that we talked about a little bit about before. So by all means, go back and revisit that. CD226, right? That is on the surface of something called natural killer cells, T cells, platelets. It's a receptor found on those that act as a co-stimulatory, like green light for immune cell activation. And so when that receptor on an immune cell binds with its partner on a target cell, it strongly promotes a pro-inflammatory response, right? Because it's a green light, go, right? What else about this section? Then they did some cross-tissue validation. And so what they're describing is the CSF trained model classified serum samples with an AUC value. It's just a statistical number of 0.08, or no, not 0.08, that's way off. 0.8 right here, 0.8. And serum trained model classified CSF samples with a AUC of 0.71, which is pretty good for the model that they were training on. So it suggests that KLS produces kind of a proteomic signature that is detectable in both compartments, CSF and serum, right? Then they used the machine learning and the classifiers to see if it can predict if a case was in episode or not, right? And so in the CSF model, they had some predictor scores, and in the serum model, they had some predictor scores. And so what they found was that proteomic abnormalities or changes were most pronounced during active episodes, and persist a little bit, and to some degree in between episodes, which may suggest that there might be some baseline dysregulation systemically or outside of episodes, but upregulate more or downregulate more during an episode. All right. And so they go on to describe the top proteins involved in differentiating in episode versus not were ones that we talked about before, right? So TGF beta, DKK, IGF-1, JAM, those were more predictive in their models of distinguishing or differentiating between an episode versus out of episode. And so their goal here, or the main takeaway with the machine learning, is that they demonstrated with the machine learning that KLS has a reproducible proteomic signature in patients, at least compared to controls, and that there's certain signals amongst certain proteins that are more measurable in episode versus out. Mapping disturbances to specific brain regions, right? And so is this happening all over the brain, or is there some sort of enrichment in certain regions? And so so that's what they intend to mean by mapping, right? And so the tissue enrichment analysis described previously and the methods to differentially reveal that differentially expressed proteins in the CSF are predominantly brain derived and less immune derived. Specifically, upregulated proteins were enriched in the cerebellum in the midbrain. Remember the cerebellum? Actually, let me nerd out a little bit. All right, here's a model, if you can see me at all. So here's the brain. All right, the cerebellum is this portion here in green. In our brain, it's not green. But the cerebellum sits tucked up back here, just behind the occipital bone. And the brain stem is, if you were to hemisect this or transect it, or do a section, this up here would be the thalamus, hypothalamus area. This would be the brain stem. All right. So in order of the brain stem, it goes midbrain, pons, medulla. All right. So pons is this beer belly looking portion. So midbrain, pons, medulla. So just so you have an image or sort of description. And so the proteins upregulated predominantly were enriched in the cerebellum and midbrain, which do have function in motor coordination, mainly the cerebellum, but also arousal and sleep-wake regulation, because a lot of the sleep and circadian pathways, while it's predominantly brain, some pathways that go to and from relay across the brainstem. All right. And those proteins that are downregulated, as they described, were enriched in the pons and medulla, which again are regions that are critical for autonomic function and control. So things like breathing, heart rate, things that we don't even think about, like sweating. But again, because there's pathways to and from, there is also pathway involved in sleep regulation. Right? And so this pattern suggests that there might be a regional imbalance in brainstem, with some areas showing an increased protein activity while others decreased. But because there's pathways that disperse amongst to and from, the brainstem is well positioned to globally dysregulate other portions in the brain, which has been consistent with imaging studies described below, or not described below, but described before. So I did a video on taking together all of the imaging studies that have been described, in which it could be consistent here. There's a widespread, generally hypoperfusion seen on those imagings during episodes. And so the key takeaway there is that the proteomic disturbances or signatures map to certain brain regions or brain stem regions, potentially linking the immune cell protein signatures that they described before with some neuroanatomy circuits that are known to kind of crosstalk potentially in sleep and arousal. And so that's a lot of stuff, right? And so in their discussion, they go on to further detail and associate some things. And so to kind of bring that narrative together, they mention here some of the top differentially regulated proteins are involved in the microglial monocyte macrophage axis, right? That activation, again, upregulated of certain proteins or cytokines, point to an activated microglia in a predominantly neuroprotective mode. And so that could propose that there's an increased neuronal excitability that could be associated with KLS episodes. And there's some other proteins like IL-34 being elevated potentially in response to or as a compensatory mechanism, somewhat limit some damage, potentially explaining why some sedatives like gamma hydroxy butyric acid has paradoxically triggered episodes in some patients, which is interesting. Despite the fact that derivatives of that drug have been used and approved for idiopathic hypersomnia. They also talk a little bit about the blood-brain barrier. And so some certain elevated proteins like JAMB, VWF, albumin could suggest that there's an increased permeability of the blood-brain barrier, which would be somewhat consistent with some sort of neuroinflammatory process. They describe that there. They also describe a little bit of the Wnt pathway, the WNT pathway dysregulation. So downregulated DKK proteins somewhat implicate a maybe disrupted Wnt pathway signaling, which again is a critical pathway for the function of synapses, and has been linked to other conditions of neuropsychiatric origin like schizophrenia and bipolar, which have been shown to share some genetic overlap with KLS, which I've talked about in a couple of previous videos. And they go on to talk about, so the DKA portion was here. There, brainstem disturbances, right? Remember that pathway enrichment analyses links somewhat immune function potentially, or immune findings related to their to specific brainstem regions, potentially providing a neuroanatomical framework for potentially localizing dysfunction, or how localized dysfunction could produce a global cortical changes that are seen on imaging. All right. Then they do a good job of talking about some of the limitations of the study, because there's always limitations to any given study. For one, this is a cross-sectional design of a single point in time, right, which kind of precludes some causal inferences. There is a relatively small sample size relative to the features. So while measuring hundreds of different proteins in a sample that is a small number of samples, you could potentially risk false positive discoveries. They have a correlated protein data right here, second by nature. Protein data is highly correlated. Proteins interact in networks that are not truly like independent of certain measurements, because there's other things that can be involved in protein measurements. This is a single time shot of the sample taken during a certain time period, right? And so this doesn't provide a longitudinal tracking across episodes, which they acknowledge. A certain episode status, right? They did acknowledge that 8 of the KLS cases, their status of in or out of episode was not known for that sample. And so some things that I think are very good about this paper, some strengths, is that it's one, novel, and it's ambitious, right? It's the first high throughput proteomic study, right? In a condition in which we don't have biomarkers. It's rigor, right? It provides a lot of strict thresholds statistically. That provides a good framework for the study. It's machine learning validation. And so training one data set and then validating it on a independent data set strengthens some of the findings here. Has great biological coherence and relevance, right? Because some of the proteins and related thereof converge on plausible pathways like microglial activations, Wnt signaling, blood-brain barrier disruption that could align with or interface with a lot of the imaging studies that we've shown and some genetic findings that they've previously talked about before. All right. Some gaps and areas that could be improved, right? The small validation cohorts, right? Their validating cohorts was a smaller sample set. The serum validation set was only 6 cases. And so expanding that out, hopefully they do a repeat study to have more of a sample set. Another big one is that I think it would be great if there was a disease control, right? And so having a control set of other hypersomnias like narcolepsy or idiopathic hypersomnia or other conditions that KLS gets mimicked for or can look like bipolar, depression, and encephalitis. And so without a disease control, some biomarkers you can't say that these are specific to KLS, right? And so things that are upregulated and downregulation, despite them showing more in a KLS sample compared to those without KLS, you cannot conclude that these are specific to KLS, all right? So those are some major pros and cons of the study. But I think it's an amazing study because it provides a launch pad for more, right? And so hopefully this provides a good foundation for readers of this study. Hopefully I was able to describe what a lot of the jargon means and therefore providing a better understanding of what a lot of these sections describe. So that's my goal for all of these videos. And so if there's any listener or watcher, so to speak, definitely comment any question, comments, concerns. But yeah, hopefully I am of help to walk through a very science-heavy paper to establish it, or to establish what these terms mean and what the authors suggest and what they mean. All right. Thank you for the listen. There's going to be more videos to come.